So in this last section on photosynthesis, we're going to be studying a phenomenon known as photorespiration, which is kind of a counterproductive to photosynthesis. And we'll see what that means right now. So your learning outcomes for this R1 is to distinguish between how rubisco acts to make or to fix carbon dioxide onto ribulose bisphosphate, and how that same enzyme rubisco, if we have too much oxygen there, can actually oxidize the ribulose bisphosphate, which wouldn't allow us to make glucose, because the ribulose bisphosphate is the starting point for fixing carbon. So we're going to be looking at different kinds of metabolisms for photosynthesis. We've already seen one, and so you're going to want to compare the functions of these, how they work. C3 we already saw, that's actually another name for the Calvin cycle. And then we're going to look at ways around photorespiration to prevent it from happening, and that's C4 and CAM pathways. Recall that from the normal Calvin cycle, there's an enzyme called rubisco, and what we were looking at was the carboxylation with the help of rubisco, where CO2 is fixed onto ribulose bisphosphate, and this is favored under normal conditions. Okay? But if we're in a situation where a plant is in very hot, dry weather, the plant, and this over here on the right is a diagram showing a cross section through a leaf. So we're looking at that cross section. We're looking inside the leaf there, and there are the mesophyll cells, which means the middle of the plant, where you see all the chloroplasts. During hot, dry conditions, these openings under the leaf, called stomata, which help exchange gases with the atmosphere, close, and that's to prevent too much water from being lost, because water moves from the ground up through the plant and then out the leaves and evaporates out. So if it's very dry, that could be not a good thing for the plant if things become drier for them. So what happens if they close that off? Photosynthesis will continue, and then we start to build up oxygen in the tissue. But with the stomata closed, you can't get rid of the oxygen, and you can't take in any more carbon dioxide. So what happens is, overall inside the leaf, you get a greater amount of oxygen, and oxygen loves to compete with the same active site as carbon dioxide on that enzyme. And so what happens here is that during this photorespiration, when there's too much oxygen because the stomata are closed, the enzyme with the oxygen present, too much oxygen present, causes the oxidation of that ribulose bisphosphate. So breaking down this ribulose bisphosphate, it's not going to allow us to make glucose. So again, this situation is going to happen whenever we're not able to get more CO2 in, and we're not able to get rid of the O2 being produced. So we're going to end up with a high concentration of O2 in the leaf, which causes this photorespiration. So there's the diagrams again of the leaves with open stomata, and on the right with closed stomata, and you can see how they don't allow, when the stomata are closed, does not allow the oxygen and carbon dioxide to be exchanged with the atmosphere. So the types of photosynthesis include C3, which this is normal. C3 is the Calvin cycle, and this is it's called C3 because the ribulose bisphosphate, bisphosphate is it's a 5-carbon compound, and then when we add the carbon dioxide, that adds another carbon, which gives us a 6-carbon compound, and then it immediately splits in half, which is going to give us 2 3-carbon compounds. So that first molecule you get after fixing carbon is a 3-carbon compound, so we call it C3 photosynthesis or metabolism. As far as preventing photorespiration, 2 other mechanisms have evolved. One is C4 metabolism, the other one is CAM. Both are going to end up fixing carbon dioxide instead of on ribulose bisphosphate first. That's still going to be used later. But first they're going to fix it on PEP, which stands for phosphoenolpyruvate. This is something we've seen during glycolysis. The phosphoenolpyruvate is a 3-carbon compound, kind of like pyruvate. And if we add a carbon dioxide on there, 1 carbon, That's going to give us a starting compound right after fixing the carbon dioxide, or fixing the carbon. We're going to get a 4-carbon compound, and it's not going to be split in 2. So we start off right after we fix the carbon, we get a 4-carbon compound instead of a 3, as in the Calvin cycle. Now this is going to be done with the help of phosphoenolpyruvate carboxylase, and this particular enzyme, unlike the Rubisco, the enzyme for fixing carbon dioxide on ribulose bisphosphate, that one doesn't have as great an affinity for carbon dioxide. So when there's a lot of oxygen present, the Rubisco ends up going through photorespiration. But over here, even if you have a lot more oxygen in the area, this PEP carboxylase has a greater affinity for CO2. So even if you have more oxygen in the area, this enzyme is only going to be binding with at the active site with carbon dioxide. So the thing is, we're not going to get any oxidation occurring. Now, there is some differences here, even though both use the C4 and CAM use the C4 type metabolism. The C4 themselves are going to have this spatial solution, and that means they're going to separate within the cells where certain reactions take place. Particularly, we're going to separate away some of the process away from where there's higher amounts of oxygen. So we're going to move into other cells away from where the oxygen can mess things up. And then for CAM metabolism, this is a time solution, a temporal solution. And here, the time of day in which the metabolisms take place differs. So it's a temporal solution to dry, hot climates. So let's take a look at normal C3 Calvin cycle we've been looking at here. So if we have carbon dioxide coming in, here the carbon dioxide gets into the cells and into the chloroplasts, and then it gets fixed, just like we saw before with the Calvin cycle. The carbon dioxide gets fixed to ribulose bisphosphate with the help of Rubisco, and then we go through the process described in the prior section to produce our simple sugars there at the end. So that's normal. But remember, if we have too much O2, then the O2 with the enzyme that helps fix the carbon is actually going to end up oxidizing this and breaking down the ribulose bisphosphate, and that reduces the efficiency of this cycle here. And so what we're going to see with the C4 pathway is they're still going to use the Calvin cycle, but we're going to push the Calvin cycle away from where there's high oxygen levels. And so to do that, there has to be a difference in the anatomy of the leaf. If we look at the mesophyll, which is the middle of the leaf when you translate that word, and you look at the mesophyll of a normal C3 plant, up here you can see there's differences right around where the vascular bundles are. The vascular bundles are where water is conducted from the roots all the way up to the plant and then into the leaves. This is where the veins are in the leaf. So this is basically a cross-section of a leaf right through the veins where water and minerals are coming into the leaf. And you can see that right around there are special cells called bundle sheath cells. And it's those bundle sheath cells where the Calvin cycle will take place, what we've normally studied. And outside of that is the mesophyll. So out here is where the rest of the leaf is, and this is the bottom of the leaf down here, and there's where your stomata are. So the stomata closes, and we have a buildup of oxygen here in the area, and there's still a little bit of CO2. Instead of trying to fix the CO2, whatever CO2 is there, onto the ribulose bisphosphate, leave that for the bundle sheath cells that are further inward, and instead we're going to fix the carbon dioxide onto phosphoenolpyruvate to make a 4-carbon compound, which you see over here. So in the mesophyll, even if there's high oxygen levels, there's still some carbon dioxide. We're going to take that carbon dioxide and fix it onto PEP to make our C4 compound. And then we're going to move that C4 compound into the bundle sheath cells and then have that compound give up the carbon dioxide. Okay? And now that carbon dioxide can go through the normal Calvin cycle, the C3 pathway. So we actually have C4 occurring here, and then spatially away from where the high oxygen levels are, we go through the normal C3 again. So the Calvin cycle is still part of this. So this is detailed a bit more. The diagram on the right, and on the left there's a few notes here. First of all, this type of metabolism is typical of corn, sugarcane, sorghum, and other members of the grass family. Grasses tend to grow in drier areas, and corn, sugarcane, and sorghum are just domesticated wild grasses that we've used to grow food from. But you can see here what happens here. So here's your mesophyll cells where you might have higher oxygen levels. We do have some carbon dioxide in the area, and the enzyme that's going to help fix the carbon dioxide onto our 3-carbon phosphatidylpyruvate has that higher affinity for the CO2 and not O2. So it's going to help fix the carbon dioxide onto the phosphatidylpyruvate. And the first compound we're going to get is oxaloacetate, which we've seen before in Krebs cycles, so this is nothing new, the compound itself. Then oxaloacetate gets converted to malate, which is just another organic acid, and it's still got 4 carbons on there. And then the malate enters the bundle sheath cells away from where there's high O2 levels. So we get it away from there. This is spatial. And in here, we're going to decarboxylate the malate. That means we're going to take off a carbon in the form of carbon dioxide and give it to the Calvin cycle. The resulting molecule that remains is pyruvate, which we've seen during glycolysis. Then that pyruvate is going to go through changes to form phosphatidylpyruvate. We have to use ATP to do that. So here we're going to be using more ATP than normal, than through normal just C3 metabolism. But still, this is better than having our Calvin cycle messed up because of the excess oxygen. In other words, from having photorespiration happen. Using the extra cost is going to outweigh or not outweigh the benefits of doing this in a dry climate, I should say. So it's better for the plant. So again, the malate, it's converted to malate, which just gets transported in the bundle sheet away from where there's higher oxygen. And in the bundle sheet, the malate is decarboxylated, and that gives us CO2, which can then enter the Calvin cycle as normal. So C4 pathway does overcome the problems of photorespiration, but not without the cost that I mentioned already. It's going to take some extra additional ATPs to do this. But this C4 pathway, or photosynthesis, is an advantage in hot, dry climates, because without it, photorespiration would remove half of the fixed carbon, which means we would get half of the glucose you could possibly make because of photorespiration using the normal C3 pathway. As far as CAM plants, this is typical. CAM metabolism is typical of succulents. Succulents are plants that store a lot of water, like cactus and pineapples and some other groups of plants. And here the CAM stands for another organic acid called crasslucian acid. So this is crasslucian acid metabolism is what it stands for. And it's very similar to C4. So here, this is temporal. So this, rather than having a leaf anatomy like we saw with the bundle sheath cells wrapped around the vascular bundle, here we're going to open our stomata at night. So at night, if you open your stomata, it's not as hot. You're not going to lose a lot of water. You're able to bring in carbon dioxide and release any extra oxygen that's been stored in the tissues. And the carbon dioxide comes in, and we're going to fix it. And what we're going to do is we're going to fix it, fix the carbon dioxide on phosphatidylpyruvate, just like we did with the C4. And then this compound is going to go into the vacuole, and it's going to be stored as organic acids like the crasslucian acid. So it'll be converted to other organic acids. And then during the day, we can close our stomata, and we can release the carbon dioxide and then allow it to go through the Calvin cycle during the day. So over here in our diagram, we see we're going to take in carbon dioxide during the day, fix it as a C4 with phosphatidylpyruvate as before, and then go store it in the vacuole, okay, of the plant cells. Vacuole of the plant cells. And then during the day, close the stomata and then have those organic acids decarboxylate and release the CO2 and now you'll have plenty of CO2 during the day that we can go through the Calvin cycle and produce our simple sugars. So in this way, we're going to minimize our photorespiration from occurring. So that's a good thing. So if we compare C4 and CAM metabolism, we've seen that both are going to be using this C4 pathway and also use the C3, which is Calvin. But when they do these two, depends on what type. The C4, they're going to separate the two pathways, the C4 and the C3, in different cells. The C4 is going to occur in the normal mesophyll cells and then move the 4-carbon compound into the bundle sheath cells and then go through C3 metabolism. For CAM, we're going to separate the time of day that happens. We're going to do the C4 pathway at night, storing the molecule as organic acids, and then release the carbon dioxide during the day so that we can go through the C3 pathway. So that's it.